So thank you for coming. My name is Anneke Alkemade, and I create 3D reconstructions of the whole human brains, but not the way that BigBrain was designed. So what I do is I create images of the human brain to derive maps, much like Google also does it. But these maps, of course, only become interesting as soon as you start annotating them. Because if you start annotating them, you can use them for other stuff. You can use them for planning a trip, for instance, to travel to Paris. And if you plan a trip to Paris and it turns out it takes you 24 hours while you're going by car from Amsterdam, you know there's something wrong. There's some of the anatomy that you are missing, that you have not annotated yet. So in that way, you can also use that type of information. Now, how do we go about this? We want to map the brain in order to create tools for people who want to do fMRI analyses, and particularly analyses focused on deep brain structures, because we know so much about the cortex, but the subcortical areas are relatively unexplored, even though it's very crowded down there. We counted them once, and there should be about 455 individual structures there. And about 7% of these structures have been mapped. That means we're systematically ignoring 93% of what is down there. And that is a problem that we wanted to start fixing. So what we did originally is we started out using ultra-high field 7 Tesla MRI. And we did this at a sub-millimeter resolution. And we wanted to cover the entire human lifespan. Adult lifespan, so we included people aged between 18 and 80 years at the time of scanning. So that resulted in a data set with 105 intrinsically aligned quantitative contrasts, which we obtained from a single scan acquisition. And the MP2-RAGE-ME was specifically designed by Matan Khan to acquire these types of data. Now, this is an enormous data set, which is very wealthy, and we need to map 455 structures, so we need help with that. And that is one of the reasons why we share all of our data openly. So if you want to play with these 105 individual data sets, you can. You can just download them. Now, we started at 0.7 millimeter isotropic, but we already realized from the get-go that that was not going to give us the anatomical resolution that we need to map out all the individual structures of the subcortex. So what we did as well is we acquired an additional slab data set. So we scanned only part of the brain, and we did that at 0.5 millimeter isotropic, again with this MP2-RAGE-ME sequence. And this has formed the basis of our atlasing project. And what we do is we create probabilistic atlases of the subcortex, which means that we incorporate inter-individual variation. And how do we do this? Well, we create a manual delineation which forms a ground truth for our atlasing. And here you see this for the red nucleus. And what we do is we manually delineate a structure twice by 2 independent researchers. And we do this to prevent or at least map out the biases that may occur in this. And we started out doing this in 105 subjects. Then we calculated how much time that cost, and we realized that it would take over 12,000 years to map out the entire cortex. So we needed a plan B, and quickly. And what we do now is we no longer delineate in 105 subjects, but we delineate in 10. And based on those individuals, we train an algorithm called MASP, which can now delineate over a total of 35 structures, which is 65 in total if you go separately for the left and the right hemisphere. So if you want to use also these atlases or the algorithm that we created, you can also download that. and use it on your data, but you need to go and validate whether our systems work properly on your data. And for that, we share detailed description of the delineation procedures we use. And we also give some example data sets. So I would advise you to go and use that if you're using atlases. But we are like the caterpillar that never has enough. Even at 0.5 millimeter resolution, we think we are not getting the anatomical resolution that we need. So we want to get to smaller structures. So we decided to also develop a post-mortem approach. It's not as detailed as BigBrain, but that is because our goal is different. We're trying to create a bridge to the MRI. And we do that by scanning whole heads, ultra-high field resolution again. I think this is 0.4 millimeter isotropic. We then perform the autopsy. We cut the brain. We create block face images, which we can use to 3D recreate things. Then we do histology, but also immunohistochemistry. And we can extract features from the reconstructions. So this is then the high resolution contrast you end up with in the MRI. All credits go to Nick Weiskopf and Evgenia Kirillina's group at the Max Planck in Leipzig. And then we take out the brain and we start processing them. And you immediately see that there is already substantial inter-individual variation. And that is something we would like to capture post-mortem as well. So what do we do with these brains? We cut them. And we cut them in a different way than paraffin. So we freeze the brain and we cut on a macrotome. We cut 200 micrometer sections. And they roll off our tissue block like this. And now you're going to have to trust me that we can actually recreate proper sections from this. Now, while we are doing the cutting, we take these block face images. So every time a section is cut, we take a picture. And this is the reconstruction of those pictures. And this is not a registration. This is just the restacking of the data. Because the photo is always in the same position, it works like a charm. If you look carefully, you can see there's a darker gray line here. That is the thickness of one section. I accidentally leaned in and cast a shadow on the photo. My bad. So what we have then is we have all these weird little tubes that have a single section all crumbled up in there. And these sections we put on glass slides. And we have in our administration the correspondence to the block face image. And that makes our registration from a 2D, 3D problem to a 2D, 2D problem. And that makes Pilou, who does the reconstructions, very happy. So what he did is he matched the block face and the staining, and then also registered to the corresponding neighboring sections, creating a 3D reconstruction of the whole brain. And that is the system that we're working from. And that is what we can use to create more atlases. And the advantage of doing 200 micrometer thick sections is that we can actually work in relative warp speed. We do about 1 or 2 brains per year. And we've started out doing classical histological stains. So we had a Bilshowski and we had a niselethionine stain. But we also included stains that would give us additional contrast to delineate subcortical structures like calcium binding proteins, parvalbumin, calretin, calbindin. And we are venturing further into that. And we have now also created a system to map out the vascular system. So on the left here in brown, you see CD31, and that's a vascular endothelial marker, so that will give you all the vasculature in the brain. And on top of that, we have SMA, which is smooth muscle actin, and which gives you information on the arterial component of the vasculature. Unfortunately, we do not have a strictly venous marker that we can use. So the venous component will be the arterial vasculature minus, or sorry, the total vasculature minus the arterial vasculature. And this has led to a full reconstruction. Let's see if it wants to run. No. Please play. Thank you. So in red, you see the reconstruction of the CD31. So that is all the capillaries included. And in green, you see shining through this SMA marker, which is the smooth muscle actin. And I think these type of data can funnel into how you want to model your bolt signal, for instance. Because if you look carefully, especially subcortically, there's something wrong here in the striatum. But you can see that some areas show a higher vascular density as compared to other areas. And that is something we don't take into account at all, from what I understand right now in functional MRI analyses. And you can also see that specific structures, such as the locus coeruleus, are just extremely vascularized. I'll just wait until it finishes, because it's just pretty colors. I can't wait. So we are using these data now to map out also smaller structures that we can't get to in vivo. And we're doing quantitative comparisons also between the histological approaches and the post-mortem MRI for validation. And what you see here is a 3D reconstruction of the individual nuclei of the thalamus. And we've based this on the calcium binding protein imaging. And I must say that if you use the calcium binding proteins, the VIM is not that easy to detect. So we went by the atlas of Mai et al., of Mai and Paxinos. So with that, you can use parcellation, you can use postmortem data for MRI by creating parcellation tools, and you can perform validation on structures that are visible both in vivo and postmortem. We are, of course, not sitting still. So the ongoing projects are the mapping of the additional architectures that we can start tackling in the brain now. And those include as well neuropathological alterations. We've also created a reconstruction of p-tau and amyloid beta. And as you just saw, we also created a vascular reconstruction. So with that, I want to thank this enormous group of people who have been working mostly on the parcellations. And I'll take some questions if they allow me.